In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum. How are you, students? I hope you all are feeling well and doing your studies with Pakistan International School Taif's virtual learning classes. This is grade first year biology, and in today's lecture number 42, today's lecture number 42, we will see about fungi topic in chapter number 7, that is about protists. So let's get started. So dear students, you can see different varieties of fungi. So today we will talk about kingdom fungi. Let's see its position in the three domains. As we have studied domain archaea first, then domain bacteria, and then eukarya. There is Leuca, like common ancestor is over there. Then we are here. Now we have seen kingdom protista. All of these we have studied here up to like this. And today we are on kingdom fungi. Now see what is fungi. Fungus or plural is fungi. So fungi is a general term is used to describe a group of eukaryotes. So here again there is a group which are eukaryotes, like they are having true nucleus true nucleus in their cell, as well as what is speciality of that absence of chlorophyll and the presence of rigid cell wall that is made up of chitin. So as you remember that in plants, cell wall is made up of cellulose. In bacteria, cellulose is made up of peptidoglycan. Here the cell wall is made up of chitin. We'll see in the next slides also. So they are in both of the forms, unicellular as well as multicellular form, like in unicellular yeast, which we use for baking purposes, and molds also, different type of molds which are multicellular. We will see in the next slides, inshallah. So let's talk about their general characteristics. So fungi are not plants. Although they are having cell wall, they are sessile, or we can say non-motile. They are eukaryotes, but they are non-photosynthetic. That's why they are not plants. So they are saprobes, like saprophytic mode of nutrition they are having. It means they live on dead organisms. So you can see here one typical structure of conidiophores of um, fungi. So fungi is having absorptive heterotrophic mode of nutrition. It means they digest the food. So for digestion, digest food first, then absorb it into their bodies. So for that, they release digestive enzymes to break down organic material and their host. So what they do, they break down that organic material on which they live, and then the food is dissolved with and digested with the digestive enzymes, and then they absorbed by their root like structures. They are called hyphae, and so they absorb the food which is dissolved by their digestive enzymes. So this is a common bread mold. That is the structure, like fungus is gonna produce at very rapidly onto this bread. So that mold is also what we can say, that is the fungi, mycorrhiza. So its food is stored in their body in the form of glycogen, like animals. So they sometimes behave like animals, like they store their food energy in the form of glycogen, like humans stored food also in the form of glycogen, but actually they are uh, very, very different from the animals. We will see in after some time. So they are important decomposers and recyclers of the nutrients in the environment. So you can see the multicellular mushroom, like some are multicellular, except unicellular yeast. So yeast is unicellular. They lack true roots. They lack true roots, stems, and leaves. So what type of structures they are having? So you can see it's stem-like, but it's not stem. So it's a fruiting body, but it is exactly different from plants. They are having cell walls, but it is made up of chitin. So chitin is a complex polysaccharide that is also present where? Yes, on the insects and crustaceans body, like exoskeletons. So their body is called thallus, as you can see, that is thick enough and grow as microscopic tubes or filaments. Those are called hyphae. So the structure which you see, that is not the singular, that is so we can say the thousands of millions of these hyphae-like structure, they get together like this and they form this thallus-like structure. So some are edible. If you see these mushrooms, these are all fungi, so they are edible, but some are poisonous too. They are best grown in a warm and moist environment. So the study of fungi is called mycology, and the scientists which study fungi those are called mycologists. Fungicide is the chemical which is used to kill fungi, like you can see the structure of the leaves. So that is parasite also, so that destroy the whole crops. So fungicides are used, the chemicals for destroying or killing fungi. So most fungi reproduce asexually, but sexually they also produce, but the method is spore formation. Asexual production is most common method and produces genetically identical organisms, as well as fungi reproduce sexually also when conditions are poor and nutrient are scarce. So we see the structure of fungi, so that is made up of a hyphae, lots of filaments. You can see these structures. So these are filaments, those are called hyphae, and collectively they are called mycelium. So cell wall contains chitin that is also present in insect and crustaceans as an exoskeleton. So plant cell wall contains what? Yes, they are having cellulose in their cell wall. And eubacteria cell walls, they contain peptidoglycan, yes. Peptidoglycan. So in bacterial cell wall, that is made up of peptidoglycan, and plant cell wall contains cellulose. So that's why fungi is totally different from these two. So its reproduction is asexually, like genetically identical, we have seen before, and some, like most fungi, are haploid throughout their life cycle because they reproduce asexually. So in sexual reproduction, they occurs when hyphae are of different mating types, like we say them mating type, not male and female, we say them positive or negative, plus or minus. So they meet and fuse together and form zygote. Some members of kingdom fungi, they are molds and mushrooms, and very common and grow rapidly. So first fungi appeared on the earth about 430 million years ago, and in the past biologists grouped them in the plants because both are having cell walls and they appeared rooted in the soil. But the roots are and cell wall is totally different from the cell wall. So later on, the unique features of fungi indicate that they are in separate kingdom, not in the kingdom of plants and not in animals. So fungi as a separate kingdom, if we see that differences from the plants, they have one nucleus in the plants, but they can have the several nuclei per cell. Plants are autotroph, but they are heterotroph and they do extracellular digestion. Starch is the main storage molecule in the plants, but there is no storage molecule, or sometimes we can say the glycogen. Most have roots, but they don't have roots, don't have leaves, and don't have stems. Instead, they are having mycelium. So they are having cellulose in the cell wall, the bigger difference, and chitin in the cell walls, but a little cellulose is also present in fungal cell walls. Though they reproduce by seeds, but fungi reproduce by seeds, those are called spores. So if we see the differences from animals, so fungi have cell wall, but animals don't have cell wall. They are having absorptic heterotrophs. Plants are also, animals are also heterotrophs, but they are not having absorptive mode of nutrition. So they are having ingestive mode of nutrition. So fungi are non-motile, but animals are motile. They can move, they can locomote from one place to another. That's why these two are having like fungi is different from animals and plants. So that is having the separate kingdom. Okay, so now we'll see it's a generalized life cycle. So it can reproduce asexually as well as sexually. So asexually, the mycelium, the hyphae, which we have seen with that is haploid. So after spore producing structures are present like sporangia, there can be conidophores, there can be asci. So they like different fruiting bodies are there. So they produce spores, and then spores are just spread via some source like water or air or some insects. So they germinate and then form again mycelium or the same structure. So they are all haploid and similar in the structure. As well as they can reproduce asexually by budding. Can you give any example of budding? Yes, like in the yeast, unicellular organism, unicellular fungi, then they can have the fragmentation also, like this asci, which we'll talk later on. They can be formed in the segmentation, and they can from fragmentation, sorry, they can grow into the normal one. And the third one is a spore. So we can see the first one is a spore formation, the second one is budding, and the third one is fragmentation. Both so these three processes are asexual reproduction. So if we just see sexual reproduction, so from mycelium, plasmogamy, like occur, fusion of cytoplasm of two types of mycelium, plus and minus, like positive and negative, that are different strains. So they fuse the cytoplasm. First of all, cytoplasm fuse, that's why it's called plasmogamy. Then dikaryotic stage comes when in one cytoplasm, two nuclei are present. So later on, the nuclei also fuses, that is also called karyogamy. Karyo means here nucleus, here plasma means cytoplasm. So then diploid stage has come in which both of the nuclei, positive and negative strains, they just get together and they exchange their genetic material. After that, meiosis is done and spores production. Again, spore production is done in which lots of spores are produced with n number. Here 2 and that is changed by meiosis into n number, and then again spores are formed and they germinate and myceliums are formed. Now I'll say its classification, in which we will see the three classes of that fungi, that is Zygomycota, Ascomycota, and Basidiomycota. So let's talk about first of all Zygomycota. Zygomycota is also called conjugating fungi, like they form conjugation between two bodies or two structures. So that is the common example of mycorrhiza, which is bread mold, and which just like which is formed on that bread, and that is called bread mold. Zygomycota, zygo word comes from zygote. Like in this, two strains, positive and negative, just join and form zygote. So that's why it is called conjugating fungi, because they form union, they form join here. So these conjugating fungi are also called zygomycetes. Zygomycota or zygote fungi is a division of fungi. It is also known as conjugative fungi also, and mostly terrestrial in habitat. So Zygomycota have two classes, Zygomycetes and Trichomycetes. So here we'll talk about Zygomycetes. So that they reproduce both sexually and asexually. So this is the structure of Zygomycota. You can see this is the bread mold. If you see, it is having in asexual reproduction the structure called sporangium. It forms sporangiospores, and that spongiospores make spores, and these spores just sit over the anywhere near about that sporangiophores. Like this, you can see spores is forming and they just grow over there, and again the whole body is formed. The rhizoids anchor the molds and release digestive enzymes and absorb food also. These are the rhizoids, so like they act as the roots, but they are not true roots. And stolons is the connection of fruiting bodies like this. So here sporangium is there, and this is called sporangiophore. It means that is the stem-like structure. So they all are true fungi and produce cell wall contain chitin. So their somatic phase as mycelia hyphae are generally choanocytic. Choanocytic means they lack cross walls in their hyphae. If you see, this one is hyphae and it is having nucleus. So there must be cell membrane, or they are divided by cell wall, because each cell has to be separated. But here these are not separated, they are just like this, and it is having lots of nuclei in their lining and in the cytoplasm, and that's why they are called choanocytic. They lack cross walls. These are cross walls, so they lack this one. So that is choanocytic. Gametangial copulation is the type of sexual reproduction in this Zygomycota. So these gametangia fuses with each other and lose their identity and develop a zygospore. Here is sporangiophore as well as zygospore. Also, where gametangia copulation is there, like gametangia of positive strain and gametangia of negative strain. We will just see in the life cycle. Then most reproduce asexually by producing sporangiophores, and chlamydospores are another type of asexual spores differing from sporangiophore that is also present in that. Let's talk about the life cycle of Zygomycota. Here Zygomycetes, or we can say mycorrhiza, that is the example of that Zygomycetes. So you can see here, we'll start from that first of all, we will see it's a sexual reproduction. So in that, we'll see from here, we can see that when the spores germinate, so they grow from every spore, mycelia grows. When mycelia grows, you can see here spores, and then they grow in mycelia. So mycelia, they depend on the spores. Okay, environmental conditions also, they are going to change into positive mating type or negative mating type. So these two sporangia, then they mate here and they form gametangia, like you can see here. Well, these are the gametangia, and with haploid nuclei. So nuclei is haploid here, both of them haploid for positive and haploid for negative mating type. Then plasmogamy occurs. Plasmogamy means fusion of, you can see here, fusion of cytoplasm just. So young zygosporangium, and which a heterokaryotic is there. Hetero means lots of nuclei over there, but just cytoplasm is fusing with one another. So here you can see karyogamy occur. You can see this one. Now it becomes one zygosporangia, and in which lots of nuclei are there. So after karyogamy, it looks like this when now diploid nuclei are there because nuclei fuses together. Now after this, this diploid nucleus or this gametangia again starts to reproduce meiotically, and like 4 haploid cells are formed again. So like in this, now it will grow bigger and bigger, and this spores are gonna form. So after meiosis, there is mitosis, like the number of spores are gonna rapidly increase. And then lots of, from here, you can see sporangiophore is formed. Then sporangium is formed in there. So lots of spores are formed. Like these spores in favorable conditions, so they just again start this germination and they start this process of sexual reproduction. Or here, asexual reproduction can also be done, that dispersal and germination, where if only one type of mycelium is there, so that is grown into a hyphae or mycelium, and then it will change into sporangiophore and then spore. You can see a large view of the sporangiophore. Lots of spores are there. There are millions of spores over there. So you can see here in this structure, life cycle of Rhizopus. That is Rhizopus is the bread mold. And separately, this is a sexual zygosporangium without one zygospore, with one zygospore. One zygospore is there. It means like cytoplasm are uniting, and now there is, we can say the nuclei are also joining. So they form the zygote here. Here we can see asexual reproduction. So asexual reproduction up to, like, sorry, not this one. This is asexual reproduction where the sporangiophore or sporangium with lots of spores inside are gonna produce. And this is so easy, like the spore formation that germinate, then again spore formation is done. So here in sexual reproduction, mating occurs. Now we'll see Ascomycota. It means sac fungi. Ascomycota, you can see its structure. That structure that is called ascus, singular ascus, and lots of they are called asci. That's what's called Ascomycota. So mycelium is septated and branched except in the yeast. So let's see here. This is mycelium. So if you see, that is septated, like from here. So that is septated, like there is a small septum present in between them, so that they can nuclear of this will be separated. So complete absence of flagellated cells. There are no flagellated cells in any of the uh, like period of in the life cycle. So cell wall contains large amount of chitin, but some cellulose is also present. So this is repeated. So harmful or parasitic in nature, and they cause several diseases in plants and humans. So they are saprophytic as well and may produce useful enzymes and vitamins, and as well as recyclers also, recyclers of the nutrients. So here we will talk about some types of fruiting bodies in all the fungi. Like if we see this one, sporangia, we have talked about in Rhizopus, in Zygomycota. This is Eschai or Eschus, singular Eschus, in which the spores are present in this Eschus. So that is in Ascomycota. And if you see, this is Basidia, that is present in Basidiomycota. So if we see it's sexual reproduction in Ascomycota. So Ascomycetes are a septate. So a septate, you can see its hyphae is having, you can see this one, that is septate, like cross walls are present there. So this is opposite to coinocytic or coinocytic hyphae. When cells of opposite mating types meet, they form dikaryon hyphae, like we have seen in Rhizopus. So Ascomycete hyphae can grow quite larger and for the time long as dikaryons, like they keep their nucleus separated, but their cytoplasm are united. So that is called plasmogamy. And after that, karyogamy occurs. So dikaryon forms a fruiting body that is called trifle or morel. So that is like that hyphae is called morel or truffle. So at the tips, each hyphae is called fruiting body, that is called eschus. Like here we can see they are sky, plural is sky. So in the eschus cell, the two haploid nuclei fuse to form a diploid nuclei. First, this one is done, diploid nuclei, then which undergo meiosis, and four haploid, you can see here, first, that you can see one darker, and up to here we are, that is dikaryon, but that is called plasmogamy. Then here is karyogamy, and the nuclei are united and they undergo meiosis and they form four haploid cells again. And some species, each nuclei divides into form a total 8 haploid cells. Now you can see 8 haploid cells are formed after mitosis. So the haploid nuclei are then packaged into sexual spores one by one. You can see here the spores are there. So they form some cyst to resist harsh conditions as well and expelled from the fetal body when they find favorable conditions. So let's talk about its reproduction. So here is asexual reproduction. You can see these spores. These spores are called conidia. So these conidia are germinating and again for this mycelium. So its asexual reproduction is done by we can see conidia, and these are conidiospores, and these conidiospores like vary in conidia vary in size, shape, or maybe they are multicellular. There are no sporangia in Ascomycota, they are conidia. So these directly develop into like conidiophores. like this type of structures, conidiophores, and they again form conidia, and then they shed down and then again form the same hyphae or mycelium. So the same, this type of hyphae or mycelium, that is going again, depends on the conidia or the spores. We don't say spores here, we say conidia. They form according to the temperature and environmental conditions. So they are changed into anthridium and escogonium. So anthridium is the male part, or escogonium is the female part. When they unite, again plasmogamy is first done. You can see anthridium that is having the connection, that is escogonium, and dikaryotic hyphae. You can see this whole escocarp is like yes, in this escocarp, we can see this one portion in this whole escocarp, this whole process is going on. So ascus, after the dikaryotic conditions, dikaryotic hyphae, that is called like dikaryotic means two nuclei are present, but that cytoplasm is same, that is united. Here the diploid nuclei is formed, that is called karyogamy, and after meiosis they form lots of haploid. These are 4 haploid nuclei, and after mitosis there are 8 ascospores, or 8 nuclei around that. Spores formation is done, like some cysts are gonna form, by which they form lots for 8 ascospores in one we can see ascus, one ascus. So in favorable conditions, they just shed from there and germinate, and they form again different mating types, and again the sexual reproduction is going on. And like that, if there is no mating type, so that spore or that is changed into conidia, and this conidia grow, and again mycelium is gonna form. So asexual reproduction, we have seen that is made by conidiophore. Conidiophore, these are like the structure of that conidiophore. In this, later on, conidia are formed, and this conidia just shut down, and they just change into like ascospore. From here, ascospore can be formed, and from here, conidia can be formed. So both bisexually and asexually, they can reproduce. Here we can also see conidiophore also. And mitosis, they form spores. These are called conidia, and germination also done, and they form the mycelium. Here we can see sac-like fungi diversity, like sac fungi, that is also called ascus, so that can be the cup shape. Here you can see is ascus, in which the 4 nuclei are there, 4 cells are there. Here we can see conidiophores, and this is also, we can see the cup-like structure. So that is the sac fungi diversity. Dear students, with this, that is enough for today, because we have studied the lots of concept about fungi. And tomorrow, inshallah, we will continue this topic in the next lecture. That is about the next group of that is Basidiomycota, and some more concepts of fungi. So that's it for today. Till then, Allah Hafiz, wish you all the best.